Good morning, good afternoon, everyone. This is the first webinar in dkNET's new webinar series. As I mentioned, we will be recording these webinars, and we will then be making webinars available through our YouTube channel as they become available. And so we'll continue to upload future webinars as well to the same channel. And this week we are doing a introduction to dkNET, focusing more primarily on the tools and services around research resource information and the associated services that we have with that, and we'll also talk about some of the other areas of the portal, and we will also be having some follow-up webinars on some of those areas as well. So I will jump right in. Oops. All right. So again, the focus on today's webinar will be on sort of the research resources, how we're identifying them, the resource reports, also how we use information on these resources to help people with authentication plans through reproducibility reports. And then I'll touch briefly on fair data services and the Hypothesis Center, both which have, both of those have upcoming webinar dates focused specifically on those areas. So what is dkNET? dkNET was launched really to help serve needs of investigators, in areas of science related to NIDDK at NIH. And what we're trying to do is provide access to a diverse set of resources, including data, information, material, information on funding opportunities, to help advance this research. And in this phase of the project, what we've really been focusing on is providing specific tools to assist researchers in doing better science, but also in supporting the science that they're doing. And so this is, the whole idea of providing these tools and services in support of rigor and reproducibility, built around the use of research resource identifiers, and I'll describe all of that. And so, again, dkNET 3.0 is the software release that has these features, and what we're currently in is we're in a public beta phase. And so the official launch of the initial 3.0 release is in early April. And so you can go to the site now, you can try, resource reports, reproducibility reports, but I do want to let you know that, since we are in a public beta, we are, adding new features and improvements, so you'll see frequent updates and improvements up until our formal release. So a lot of the tools and services that we provide rest on an underlying ability for dkNET to aggregate information across a number of different data and information resources. So the Discovery Portal, which is one area of dkNET 3.0, basically allows you to search across 200 different databases and also PubMed to discover information, data sets, resources that are useful in your research. And for example, what you see on the screen now is that there's this search within dkNET, and then you can go into and see, for example, what the NIDDK central repository has in this in the context of available data. And this is fully open, and there are also some back-end programmatic interfaces so that you can also build these types of searches into your own tools. And so a lot of what I'll be showing in future slides rests on our ability to be able to aggregate this type of information from across a large number of sources. So one of the issues, and this is where we start talking about sort of research resource identifiers, is that key biological resources are not easily found or identified in the literature using sort of the standard way to cite different resources. And so there's publication in 2013 that described this. This is just an example of within a paper looking at how someone described a mouse, trying to find it then at Jackson Labs, but you can't actually through their site find it, and there's no easy identifier to use to be able to try and track down what organism this was. And so this is where the advent of RRIDs come in. And so research resource identifiers are unique identifiers for research resources described in the method sections of papers. They're meant to be search friendly, and they're provided by a number of different independent community registries that track single types of resources. So for example, in the case of antibodies, the example on the right, there's the antibody registry, which provides identifiers for millions of different antibodies that are used in the literature, and you can use these then to track down specifically to the vendor's information on that antibody. And so this is just an example of a paper that used dkNET to actually help find the RRIDs for their experiment. Currently, RRIDs are supported by many major publishers. There are now more than 700 journals that have RRIDs appearing in them. And information on RRIDs are included now in a number of different author guidelines. So if you're publishing papers, you may be getting already information about requirements to provide RRIDs for your different resources described in your methods. And so there were two main reasons for creating these resource identifiers. One was in tracking. So for example, who is using this same mouse? So if you're doing research using a specific model organism, that you can easily find additional researchers, additional publications around that model organism, for example. The other area is in terms of rigor and transparency. So when one looks at authentication of various resources, one would like to know how these resources have performed in the past, and the only way to do that is to get information, to pull that information from the literature. By having these resource identifiers, that makes it much easier for us to pull that information from the literature and provide that to researchers to assist them, when they are constructing, their studies. So through an initial pilot project, RRIDs were brought into a number of different journals, and there was a follow-up paper in 2015 that looked at differences in identifiability once RRIDs were introduced, and also what are the for example, costs to curation and ability to discover these resources. And so in terms of identifiability, having these identifiers provides a simple way for anyone to track these resources through the literature. So building on that, now that we have a foundation for tracking various resources, organisms, antibodies, software, we can now start aggregating some of that information. And so that's what dkNET's new resource reports are meant to do. So what we are are doing is providing now a simplified overview report for any of the resources that we are able to track and provide information, for example, on who is using them. Do we know of any issues? One of the features that we will be bringing in a future release is how do they compare? And for example, since we know, for example, who's published now with these various resources, you can even try to find collaborators near you, that can provide potentially additional information or assistance with the resource that you're looking at. And so when you look at the page, there's very basic information in terms of the specific type of resource. So here we have information specific to an antibody in the little information section. Then below that, we have the usage and citation metrics, where we provide information, again, where this resource has been found in the published literature, and also then the ability to search for a collaborator network for researchers near you. Again, when you look at the resource reports, right now we have 4 different types of resources that we provide these reports on, which is tools, cell lines, antibodies, and organisms. We will be adding some additional resources in the near future as we build, as we build this out, over the project period. So when you get to looking at the resource reports, in terms of the search, you end up with a search page that allows you then to do a resource-specific search. So, for example, in antibodies, you also have the ability to add specific information about the vendor and catalog number to make it easier for you to find the resource you're looking for. Once you have that, you can select the resource that you want to look at, and you can do that either in the snippet view, or we also have what's called a table view. And so the table view provides sort of an easy way to compare, a whole set of search results in a tabular fashion, making it easier for you to scroll and compare the various values, that are returned. So once we get this the summary report, we can then look at the usage, and so that's a sub-report from the main report. And the resource usage gives you information, again, on the publications it's been found in, and then also the articles, over the years that it's been found. So like how many mentions in various years. And you can then also see information on other research resources that are mentioned with this resource. So this is the idea of finding resources that are related to this specific resource. And the co-mention piece of these resource reports is actually undergoing a revision right now, so there'll be some additional features in the co-mention section, coming shortly. For the Collaborators Network, once we have the ability to see which publications they're in, PubMed also provides some of the location information, the affiliation information for researchers, and then we're able to use that information and geocode that information so that we can actually do searches around institutions, locations, so that you can find sort of a list of researchers that have used a specific resource around your location. And so in this case, we're looking sort of in the region, sort of statewide, so California, and seeing how many people sort of that we know about through what we've been able to extract from PubMed, are using this antibody. And again, we aren't able to get potentially all the information about these affiliations because we are pulling this information from PubMed, so we're limited to the specific information that that PubMed provides for us to be able to pull this information. Now, one of the things that we've been adding to these resource reports are ratings and alerts. So there are a number of sites that provide information. So, for example, for antibodies, ENCODE, the Human Protein Atlas, and a number of other resources provide information on antibodies. The ICLAC, which is the International Cell Line Authentication Committee, provides information on problematic cell lines. And then in regards to software, there are a number of software projects that provide a repository and registry of software resources and have feedback as well. And so what we're doing with this information is we're trying to aggregate as much of this information that we can find from these various sources, and then make that available to you through these resource reports. And so, for example, in the middle of the screen, you'll see a ratings and alert pane with the red text. And so, for example, here we have information from ICLAC on a problematic cell line, one that was misidentified. And so we try to make that as visible as possible on these resource reports. We also have additional information on validation data. So, in terms of ratings of antibodies, a lot of sites, don't provide, a rating, but do provide further information about validation. And so we're taking this validation information and also making that available so that you can link into, that information and take a look at that from different places that provide that. For the individual resources, again, all these resources that are identified through community registries, and these community registries, all have sort of different places where one adds that information. So, for example, if you have an antibody that you cannot find, you would go to, in this case, the antibody registry, which allows you to, add an antibody, even a personally developed antibody, and then get an RRID provisioned. And for the antibody registry, for example, within 1 to 2 business days, you'll have, an RRID for the antibody that you're using. On this screen, you also see that, for example, we've also been working with AdGene and the National Center for Bioinformatics at NIH, in terms of adding new types of resources. So from AdGene plasmids and from NCBI Biosamples. So as I mentioned earlier, a couple slides ago, there are additional resource types, that we will be introducing, over the next, few months. And just to give a little information on how this works, and so we at dkNET, have, for example, with Biosamples, we've worked with the Research Resource Identifier Initiative and the Integrated Islet Distribution Program, to help find, a way to, uniquely identify their biosamples. And so we coordinated meetings, with the biosamples databases in the EU and US, to help provide these identifiers for, in this initial case, was for pancreatic islets used by the IIDP researchers. And so, as, new resources become available, we work with, these community registries to try and assist, in terms of getting, the RRIDs set up that can then be used, within the publications themselves. So the resource reports sort of provide then a a background and the support needed to develop further types of reports to assist researchers, in the various tasks, that they need to complete, in terms of, for example, submitting proposals to the NIH. And so this is the section around reproducibility reports, and really focusing on authentication. So what, is needed to do, when one authenticates a cell line. So we know that, there's, issues with publications of contaminated cell lines, right? So we all, when doing research, with cell lines, that's one of the key key aspects of authenticating a cell line. But even though that is sort of something that should be done for all cell lines, people are still publishing papers with contaminated cell lines. And so this is a paper, from Science. And in a recent paper from Babak et al. That was just published this year, looked at, RRIDs and how they impact sort of the reported use of problematic cell lines in biomedical literature. And, in using, RRIDs in papers, that are required, we do see, sort of less problematic cell lines when people are using, the RRIDs. And that is at the publication phase, right? So this is when, you're publishing a paper, and suddenly, potentially, you see that, when you're getting information on the RRID, that this alerting information comes along with it. And so, one of the issues is that we like to know about any issues or any problems, or any specific requirements around the use of resources, much earlier on in the scientific process, and that's where the use of RRIDs and these sort of warnings and alerts that we're starting to bring in, can be used. So, for example, if you do have to, find an RRID, earlier on in the process, you will actually get information then about problematic cell lines, or potential issues, with an antibody or with a software tool. And so basically, these warnings are great, in terms of, publishing requirements, because, that is one place where, we definitely want to know, if cell lines or antibodies have issues, before they're, put out in the published literature. But, we should try and know this, or provide this, for researchers, so that they can get this information earlier on in the process. And so this is where, we started looking at developing tools to help, researchers when they have to provide information to NIH for the authentication of key biological and or chemical resources. And so, through dkNET, we can now assist researchers in preparing, their authentication plans for cell lines and antibodies. And for example, in terms of how to authenticate antibodies, there's, the Nature Methods publication that has, the proposal for validation of antibodies that provides a number of different validation pillars, and all this information that's out there, in the literature for this, we can provide in a sort of a simple and direct fashion. And so through, the reproducibility report section, we provide information, for example, on the main page without even, adding any information about resources that you may be using. We already provide some of the information on, the antibody authentication plans and those pillars, so that you can have that information. We also link to some of the information from NIH. So providing an information resource for people, about developing these authentication plans. However, if you want to take this, a step further, you can actually start adding your resources and develop a report for yourself about, the research resources, reagents that you plan to use in a study to help you actually, provide information for, these authentication plans. And so within the reproducibility report, once you log in, you're actually able to create a report for each experiment. So each experiment basically becomes its own collection of items that you wish to add. And so, for example, here you see, one antibody and 3 cell lines that have been added. When you actually, once you've created a report and start adding information, we will actually then highlight, for example, if the cell line that you're preparing to use or the antibody that you're preparing to use, if we have found any information about that that is problematic, we actually highlight that for you, again, as visibly as we possibly can within this report. And when you actually then generate the report, we basically then provide information. So on the left, what you'll see, sort of our report of the resources that you're using. In this case, it first goes through the cell lines, so we'll provide information on cell lines, where we're getting the information about these cell lines, provides a table of the cell lines that you've actually included within your report, and then gives you information on authentication plans for cell lines. So in case of cell lines, basically the short tandem repeat process is what is used. So STR profiling, and we then provide you the links to further information about those techniques in the sources section down below. And again, within the table, we also provide information, about the resources. So, in all steps of this process, we're trying to alert researchers, to, any issues or warnings or alerts that we may know about a resource, so that we can catch this early on, so that, time, money isn't wasted, using a resource that is known to have issues. In terms of antibodies, so this is the antibody section of the report. So again, we provide information on, the antibodies that you selected, again, where we're pulling this information, and then we have a validation section. And here we provide information about, making sure to check the target organism. So, for example, the antibody registry gets information, about the use of antibodies, and for some of this information, you can actually check directly with what's in the antibody registry. But then there are also, suggested validation methods from the pillars, I mentioned before, that you can, also put in your authentication plan. For example, also in terms of the genetic pillar, in this case, looking at model organisms, we can actually also link to, dkNET's information on model organisms. And in this case, we can, provide the ability for researchers to also find a related model organism, in terms of their validation of an antibody. And then at the end of the report, we basically provide an index of all the information that's known that we have about a resource. And this is then pulled directly from, again, these external resources that we're linking to. So basically we provide, this report that you can then save. And go back a couple slides. So you'll notice that there's a snapshot report button on the report page, and this allows you to save a report at multiple time points. So, for example, if you've begun an experiment or you're about to begin an experiment, you can collect all your resources, check, if we know about any issues with a resource, but also look at, potentially other papers that have been using this resource as you plan your experiment. You can snapshot that to get a picture of your resources at that point. 6 months, 9 months down the road, you can actually come back and look at your resources and see, if, there have been any warnings or alerts, that have, been registered around, the resource that you're utilizing. And you can then again re-snapshot, generate a new snapshot, of that. And you notice that in this example, there are already, 3 snapshots that have been created for these sets of resources. So you can sort of have, a set of timed reports, that you can always go back to, based on where you were. And one of the plan features that will roll out probably over the next half year is to have more immediate alerting services around these features. So that sort of covers the 2 new areas around the resource reports and authentication plans and services for authentication plans. I now want to sort of touch briefly around some fair data services that we're working on, and also the Hypothesis Center. Again, this is mainly to sort of foreshadow upcoming webinars, which we will be having over the next month or so. And the services that we're building around fair data really, are meant to support, these guiding principles, that around data that are now being used by NIH and many other institutes, repositories, in terms of, how does one have to treat data to ensure, that it is properly managed and stewarded, for use, by the scientific community. And so that means that there are a number of high-level principles, to ensure that data are findable, the data is accessible, right? So that, users are able to, both humans and machines are able to access it, and that data are interoperable and reusable, in the sense that, that they can be used to, do new studies or to do validation studies, be combined with other data. And these, principles are, really meant, to, assist, with the discovery and reuse of this data by third parties. Okay, so that's not the original developer of the data, the original research study that produced the data, but allowing other researchers, and not just researchers in, the domain, of science that the data was potentially generated in, but, in other disciplines. So, for example, a lot of imaging data, different types of imaging data is very useful to algorithm developers, if they're developing denoising techniques, or other techniques around imaging data. And being able to access large amounts of MRI data, for example, can be very useful, to research in fields like that. And again, this is being recognized by many major initiatives, including National Institutes of Health, the National Science Foundation in the U.S., but also in Europe, through their Elixir activities, and also the Horizon 2020 programs. And in terms of fair data resources, so right now, these are found under reproducibility support. They may be moving shortly, to their own space on the web page. But the fair data resources, provide a number of tools and data right now in terms of, helping you or helping the researcher find, appropriate repositories, for example, to place your data. And so we have an information page right now on best practices for managing research data. We have a list of community-approved repositories. If you have any questions or if there are any issues, in terms of selecting a repository, you can actually get in contact with us, and we can actually work with you to help find, a repository that meets your needs. And then, coming down the line, as a future deliverable of the project, is a tool for actually, helping create, a fair data plan that will actually help you select, the appropriate repository or repositories, for the data that are being generated, by your research studies. So again, this is, right now we have, some informational resources available to you, around generating, data sharing, data management plans, but we will be providing additional tools, as we move forward. And there's also going to be a webinar focused specifically on fair data in the month of May. And the next tool that I just want to give a brief overview of is the Hypothesis Center. And so the Hypothesis Center is an area of dkNET that's being built in partnership with Dr. Neil McKenna at Baylor College of Medicine. So he is the lead for the Signaling Pathways Project, which is a knowledge base of omics data, that has a number of data mining tools built on top of it. And the important thing here is that, this ties to, the previous discussion of fair data in that, the data that's brought into the Signaling Pathways Project goes through additional curation, from, the repositories where this omics data, is currently deposited. And a lot of this curation is really to make, this data more fair, to ensure that it is interoperable so that you can do, these data mining operations across data sets, bringing these data sets together to allow, the ability to provide, for example, what you see here on the top left is a consensome analysis, where you're listing the genes, ranked according, to a meta-analysis of their differential expression. And so this is a new set of tools, that are being provided to make it easy for researchers to explore this data and develop new hypotheses. But it relies on this data, being brought in and annotated, through these FAIR principles. And so again, this is another specific webinar where you'll be able to learn all about the Hypothesis Center and the Signaling Pathways Project. And this webinar will be, I believe, end of next month in April, if I remember correctly. And then the last thing I wanted to touch on was that, through dkNET, we're also supporting student internships to help, undergraduate, graduate research students to learn about dkNET and about some of the concepts around fair data and about, sort of the informatics, that goes into dkNET. And so we do have a summer of data student internship program, and this is a program that will run for 6 weeks. There'll be a set of webinars, online discussions, where we'll meet with the students, and then there'll be a small stipend for participating in the project. And applications are being accepted into April. And so, that sort of finishes our introductory sort of webinar around dkNET and its support for information around research resources. We have our next webinar on April 12th. And again, these webinars are going to be held every other week. If we can keep that schedule going. And next week, in 2 weeks, Dr. Anita Bandrowski will be talking specifically about the research resource identifiers that we introduced today. And so she'll be diving into that into more detail. With that, I wanted to thank everyone. And if anyone has any questions, we can take those now.